lecture to the neuroanatomy material we'll be presenting to you this year. Just for your information, this is the basic organization of what I want to do in this talk, give you a few early definitions. We're going to talk about what are called the descending motor pathways, 3 of them. Then we'll talk about integrating patterns within the motor system, and I'll give you a quick summary. Let's start. First off, before we get to the definitions, I want to make it clear what I'm trying to accomplish with this. I'm really trying to give you an overview and an introduction of this system. I am certainly not going to do adequate depth. More than anything, I just want to give you the landmarks that we will hit in subsequent lectures, so when you get to those lectures, you've got a little bit of familiarity, a little bit of a foothold. If I can give you that general familiarity, then we hope that the pace and the material of neuroanatomy that we're going to present to you won't be quite as daunting. Let's start with some key definitions then. The functional cell of the nervous system, the cell that exchanges information, is called the neuron. We have to be careful not to use neuron and nerve as synonyms. They're very different things. The cell body of a neuron is referred to as a perikaryon, and when we have groups of perikaryons together within the central nervous system, and by central nervous system I mean the brain and spinal cord, we refer to those groups as nuclei. You'll remember that a neuron has input cytoplasmic extensions called dendrites and output cytoplasmic extension that we refer to as an axon. It is a collection of axons that make a nerve. When we have a group of axons running together in the central nervous system, we refer to that as a tract. Because axons are most commonly myelinated, and myelin is really just a phospholipid, and to say a little fat, it appears white. So when we have groups of these tracts, we refer to that portion of the central nervous system as the white matter. When we have groups of perikaryons together, we'll refer to that as the gray matter. There's a whole lot of information here. Not going to worry about most of it, but I want you to appreciate that the brain is divided up into several key parts. We kind of name them from front to back. The large outer portion is called the telencephalon, and it is dominated by the cerebral cortex, where we'll start our lecture in just a few moments. Deep to the telencephalon, and the telencephalon sits on the right and left side, shall we say, deep to it in the midline is the diencephalon. The diencephalon is dominated by a large structure called the thalamus. Inferior or posterior to the diencephalon is a small region of the brain called the mesencephalon, more commonly just referred to as the midbrain. If we move back from the midbrain, we see this kind of swollen, expanded region. This is the pons, which is a portion of the metencephalon. Immediately superior, here's our pons in this diagram, immediately superior to it is the other portion of the metencephalon. This is the cerebellum. The metencephalon is continued inferiorly by the medulla oblongata, which forms the myelencephalon, and when the medulla oblongata reaches the foramen magnum, that large opening at the base of the skull, we change the name from the medulla oblongata to the spinal cord. We're going to work through all of these layers of the brain. Let's start up with the cerebral cortex. So again, the cerebral cortex is the large lateral portion of the telencephalon. If we look at it on the surface feature, we can see all these nice convoluted ridges. Each one of these ridges is called a gyrus. The little groove in between The adjacent gyri is called a sulcus. So we have gyri and sulci, which increases the surface area. If we just took a slice through our cerebral cortex here and looked at it, we can see there's two very distinct different colors. And that's because the outer surface of the cerebral cortex is all gray matter. It's our perikaryones. The inner portion, what looks light on your image here, is all white matter. It's the axons projecting away. All of the gyri and sulci of the cerebral cortex are named. We aren't going to worry about those for this lecture, but I do want to give you one main landmark. If we look at the side of the cerebral cortex, there's one large sulcus here that effectively divides at least the anterior part of the cerebral cortex into an inferior and superior portion. That large sulcus is called the lateral sulcus. And if we go up to the superior surface of the cerebral cortex, we can see one sulcus running down and intersecting the lateral sulcus. Actually, it doesn't completely intersect it. This sulcus coming down is called the central sulcus. So think of the central sulcus as running perpendicular to the lateral sulcus. Well, in the same way we've named the sulci, we named the gyri, and the gyrus right in front of the central sulcus is called, conveniently enough, the precentral gyrus. Precentral gyrus. Why do we care about that? We'll look at it on this image here. So again, here's my lateral sulcus. Here's my central sulcus. So in red, the precentral sulcus has been shown, and it's indicated that functionally this precentral sulcus is known as the primary motor cortex. This is where most of our conscious motor movements begin. You'll notice that immediately rostral to it, there are other portions of the motor cortex. The supplemental, it's often called S motor cortex, and then the premotor cortex, shown here. All right, we want to deal tonight with two aspects of the motor system. The first is a pattern of activation and inhibition, and the second is refinement and integration. For me, the easiest way to think about this is think about the movements you make in your body as you're walking down a sidewalk. Right? So we have rhythmic movements of flexion and extension of our legs, rhythmic movements of the arms and shoulders, all coordinated. We want to figure out how that happens. To do that, we need to start back up here in our cerebral cortex. If we zoom in to the very outer part of the cortex, to the gray matter, as I said before, the gray matter is full of our neuronal cell bodies, the perikaryones, and in that primary motor cortex, these perikaryones, these neurons, have a very particular shape. They are called pyramidal neurons because it's thought that the perikaryon itself is roughly pyramid-shaped. It's not really, but that's how it gets its name. We refer to these pyramidal neurons as upper motor neurons because this is where motor movement starts. Now remember, the output side of a neuron is the axon. So the axon is shown here in red. The axon projects away from the perikaryon of these pyramidal neurons, and it's the axons projecting away that form this white matter, what we call the internal capsule. We'll come back to that in just a moment. So the first part of the descending motor system that we want to talk about is the dominant one. This is the corticospinal tract. Corticospinal tract is responsible for most of the voluntary movement of our body. It starts in those pyramidal neurons we were just looking at, especially pyramidal neurons in the primary motor center. Also the supplementar and the premotor, but especially the primary. It projects all the way down to neurons located in the spinal cord. We'll talk about these are what are called lower motor neurons. And it is, again, the major pathway for voluntary movement. So let's start back up. So here we have our cerebral cortex in this dissected view. All of this material coming down are bundles of axons. These are all white matter. So we've got all of these perikaryotes scattered around. All of their axons have to come together because remember, the telencephalon is this large expanded area that covers the rest of the brain. It has to narrow down into this little group of axons here, and it's that little group of axons that will project down to the rest of the brain. This little group of axons is referred to as the internal capsule. And if we come in, we can see right here a map of the internal capsule. Now there's some landmarks here, that thalamus. All of this mass is called the basal ganglia. We'll come back and talk about this. Our internal capsule is really this large V-shaped mass here. And again, the internal capsule is just a bundle of axons both going up to the cerebral cortex and coming down from it. We can map out where all the axons are in the internal capsule, and as I've indicated here, the axons associated with the corticospinal tract occur right in this little clump. What's interesting about this is what a small portion of the internal capsule this represents. When you consider the corticospinal tract is what's taking care of all voluntary movement. We'll come and talk about what most of the internal capsule is a little later. The A, T, and L that you see here is because the corticospinal tract is very organized. So we know that the upper part of it in this image is going to the arms, the middle to the torso, and the lower part to the legs. All right, so we have the corticospinal tract descending from the cerebral cortex by way of the internal capsule. Remember, the internal capsule is going to go from the telencephalon to the midline, through the diencephalon, which is really what we're looking at here in this image, and immediately below the diencephalon is the midbrain. The midbrain is dominated by the large red nucleus. This is a group of cell bodies, a group of perikaryotes, stains in this characteristic fashion. Actually has a bit of a red coloration in the brain. And on the surface of the midbrain, on the ventral surface, we have these swollen trunks called the cerebral peduncles. These are all the connecting axons running up and down to the cerebral cortex, and again, we can map those out, and what we find is about the middle third of this mass is our corticospinal tract. We'll come back and talk about this, but note that essentially wedged in between our corticospinal tract here and our red nucleus, this light mass here is the substantia nigra, the black body of the brain. All right. We move further back, and remember, inferior to the midbrain is that swollen mass, the pons. Here we have the large, swollen body of the pons, and within that swollen body, we can see a group of axons here we've cut in cross-section. That's because the corticospinal tract is flowing right through the pons, and these are the corticospinal axons. We move inferior from the pons and we get to the last portion of the brainstem, the medulla. And here the corticospinal tracts have come together with some other tracts and they form this swollen node on the inferior surface of the medulla oblongata. And this little node here is called the pyramid. And because the corticospinal tracts make up much of the pyramid, they're sometimes called the pyramidal tracts. If we take a look at a whole brain here, you can see the large swollen pons. Everything behind it here is the medulla. You can see these large swollen masses on the upper surface of the medulla. That is the pyramid. And again, the majority of the pyramid is the corticospinal tract. Well, the corticospinal tracts don't just flow into the medulla. they cross in the medulla. And in the nervous system, when tracts cross from one side to the other, we refer to this as decussation. So I love this image here. What we've got, here's our cerebral cortex up here. This is the internal capsule. So we're looking at the corticospinal tract coming down the internal capsule into the midbrain, into the pons, and then here in the medulla, the streams cross. You can see it here kind of diagrammatically. Right? Here's our midbrain with the red nucleus, cerebral peduncle with corticospinal. Here's the pons, corticospinal coming down, and whoop, here it jumps from this side of the brain over to that side. So in the corticospinal tract, we get pyramidal decussation so that upper motor neurons on the left side of the cerebral cortex are going to drive the right side of the body. The decussation is not complete. It's about 90%. Those axons that decussate form what are called the lateral corticospinal tract. There's about 10% that do not decussate, and they form what is called the ventral corticospinal tract. Remember, the medulla oblongata, where the pyramids are, is the most inferior part of the brain. If we follow those tracks further down, we get into the spinal cord. Excuse me. A spinal cord, like the cerebral cortex we just looked at, is made up of gray and white matter, same composition, myelinated axons forming the white matter, pericardions forming the gray, but notice the pattern is opposite here. The white matter in the spinal cord is on the outside, the gray matter is on the inside. The white matter is divided into a series of functional groups we call tracts. The gray matter is divided into what we call the horns. So we often refer to the gray matter as looking like an H or a butterfly. I prefer the H. The upper two limbs of the H are the dorsal horn. The lower two are the ventral horn. We can follow the corticospinal all the way down. So here's a cross-section of my spinal cord. Here is my dorsal horn and my ventral horn. And we see that that lateral corticospinal tucks just immediately lateral to the dorsal horn of the gray matter. My anterior or ventral is down here, right beside the ventral fissure, medial to my ventral horn. Let's come back to that gray matter of the spinal cord for a moment. And in that ventral horn of gray matter, we're going to have large multipolar neurons. And these multipolar neurons are known as our lower motor neurons. So that pyramidal neuron we just looked at in the cerebral cortex that we traced through the internal capsule, through the cerebral peduncle, into the corticospinal tract of the pons, through the medullary decussation, is going to come into the spinal cord, run in that lateral, we'll make it a lateral one, into the lateral corticospinal tract, and it's going to form a functional contact, a synapse, with one of these multipolar neurons in the ventral horn of the gray matter. So this lower motor neuron is then going to carry that information out to the muscle. You'll remember from Gross the basic structure of a spinal nerve, right? We have our dorsal root ganglia leading to the dorsal root of the spinal nerve. This is our input side. Coming into that dorsal horn of gray, the ventral horn, we have our lower motor neuron, gives rise to the ventral root. Remember, these two come together to make a spinal nerve, these two being the ventral and dorsal root. That spinal nerve occurs right about the area of the intervertebral or just vertebral foramen and is going to pass out onto the muscle. We're talking about locomotion, so let's just fixate on the leg for a moment. You remember the schematic of the lumbar sacral plexus and the sciatic nerve. We had you learn all these roots, the first sacral, the second sacral. So what we're really looking at there is spinal cord levels So for our purposes, what we're going to think about are a group of lower motor neurons that are giving rise to that spinal nerve at these different levels. We aren't going to worry about the branching, the fact that we have our dorsal and ventral root. These, remember, are ventral roots. Don't care about that. Ventral rami and dorsal rami, I should say. I'm sorry. Don't want to create a problem now. But the corticospinal is not alone. Our system is much more complex than that, and there is redundancy, shall we say, in our system. So another one, if we want to understand locomotion, we have to look at the corticorubral tract. Okay? Again, this starts with pyramidal neurons, mainly in the primary motor cortex. It projects down to neurons in the red nucleus, that large body that kind of dominates the midbrain. So we can come again to the internal capsule, and now we can see this little patch of the internal capsule here where the corticorubral tract would be running. Again, it's going to come down, and its target are parakaryones, neuronal cell bodies, located in the red nucleus of the midbrain. This is a simple little schematic about what's going on. So what we're talking about here is that big black arrow. The cerebral motor cortex projects down to the red nucleus. All right. As you can see, there's a lot of other things coming and going at the red nucleus. We'll come back and talk about a little bit about that later. What I really want to focus in on here is the bottom half of this diagram, because the red nucleus takes that activity coming down from the cerebral motor cortex and essentially relays it, forming what is called the rubrospinal tract. Notice something important that's showing here. The rubrospinal tract decussates, right? It shows it on the right side crossing over to the left side. This decussation doesn't occur in the pyramids. It occurs up in what is called the tegmentum. Much higher level decussation. Behind the midbrain is the pons. We follow back the pons. Remember, our corticospinal axons are running right in here. Up on the side right here is our rubrospinal tract. We get all the way down in the spinal cord. I don't want to do too much redundancy here. We can see the rubrospinal tract located just inferior to the lateral corticospinal tract. Rubrospinal tract in man functions primarily in the arms. It's thought to be what drives the swinging of the arms when we walk. If we have an accident in the brain so that the corticospinal tract is destroyed, but the rubrospinal tract is still intact, what we can see is that the rubrospinal tract mainly drives flexion of the arms. Because when the corticospinal input is lost, the arms will go into a rigid, flexed position for a little bit. And this is an interesting thing. What it really means is that the corticospinal and rubrospinal tract are constantly kind of battling out. And it's the combination of the two that gives us the smooth, rhythmic movement of our arms. If we only have one or the other, we don't get the kind of coordinated movement we're used to. In other animals, the rubrospinal tract extends down to the legs quite heavily and plays a role in locomotion. In man, the rubrospinal tract is thought to mainly stop at the arms and not do much for our legs. That being said, experiments have shown that in other primates, if you remove the corticospinal tract, the rubrospinal tract will adapt and start taking over some of the functions for locomotion in the legs. The third tract I want to talk about is the corticoreticular tract. This, once again, starts in those upper pyramidal neurons of the primary motor cortex, and it projects down to neurons located in the reticular formation. Again, we can come down and we can map these out within the internal capsule. They're going to run very close to the cortical rubral tract. And then we get down into what is called the reticular formation. The reticular formation is this very messy mass of nuclei. They occur mainly in the medulla and the pons. You've got to get used to recognizing the pons just by this swollen area. So all of these colored masses are the reticular formation. So there's a lot of them in the medulla, some in the pons, and they extend a bit up into the midbrain. So these cortical reticular fibers are going to come down, they're going to synapse in some of these nuclei of the reticular formation, and then from here, those parakaryones in turn, their axons will form a reticulospinal tract. Lost my mouse, there we are. And the reticulospinal tract you will find right inferior to the lateral corticospinal, as medial as you can get. The reticulospinal is a very important one. The reticulospinal seems to function mainly in extension of our legs and the maintenance of our trunk against posture. Again, we can see the role of the reticulospinal quite clearly. It battles with the corticospinal, shall we say, same way rubro does. If you take away the corticospinal, and this unfortunately happens more commonly, especially like in motorcycle accidents, because the lower part of the brain is intact, the upper part has been damaged, what you tend to get is extensor rigidity of the legs and the lower trunk. And that's because these are the muscles that the reticulospinal preferentially activates, and it's still intact. If you think about this and you think about locomotion, you can understand that in order for us to take a step, we have to essentially fall forward just a little bit. So we have to deactivate that reticulospinal for a moment. So you can think of walking as a series of interactions between the corticospinal and the reticulospinal tracts. We do know that the reticulospinal tract is very rhythmic, and it is probably what coordinates the movements of our legs during locomotion. So something as simple as walking down the sidewalk is actually a fine-tuned interaction between three of these descending tracts: the corticospinal, the cortical rubral and rubrospinal, and then the cortical reticular and reticulospinal tracts. I've given you a simple mock-up of these three here. So my corticospinal is in red, my cortical rubral, the rubrospinal in blue, and the cortical reticular, the reticulospinal in green. I've indicated all three of them as starting up in the cerebral cortex. We will argue that they're all starting in the primary motor cortex, and they all do. So they're all coming down here through my internal capsule. We know they'd be at slightly different spots into the midbrain. Notice that the cortical rubral is synapsing here in the red nucleus. The others just pass through. Here in the pons and the medulla, the reticular is synapsing on these formations. We get pyramidal decussation here of the corticospinal. We get tegmental or midbrain decussation of the cortical rubral. The reticular formation generally doesn't really decussate. It's bilateral. It's unusual in that respect. The one thing all three of these have in common is that they're all going to project down to those lower motor neurons within the spinal cord. But most commonly, the final projection where the axon moves into the gray matter of the spinal cord, out of the tract and into the gray matter, the axon itself doesn't synapse with the lower motor neuron. It synapses instead with another cell located there, what's called an interneuron. The significance of this for us is it creates local circuits within the spinal cord. You've studied some of these, of course, when we've talked about spinal reflexes, and we'll talk about more of these during the neuroanatomy course. But for here, I want you to appreciate that a an axon that's coming down onto the right ventral horn, let's say it's going to the flexors of the right knee, it's going to go to an inner neuron as well, and that inner neuron may well cross over to the other side of the spinal cord, and it will inhibit the flexors of the other knee, for example. Why would it do that? Well, think about rhythmic walking. You only want one knee in flexion at the same time. I'm speaking simplistically here, but I think you get the idea. If both knees are flexed at the same time, you're going to be falling. So the same signal, ultimately, by way of these interneurons, the same motor signal that is bending one knee, that is activating one flexor, is inhibiting the other flexors. So in addition to the complexity created by the interaction of these three descending pathways, we have these spinal circuits. And the spinal circuits are mainly working on antagonistic muscles, flexors and extensors around the joints, but also the muscles on the contralateral side of the body. If this was all there was, if we only had our three battling descending tracts and then the complexity of the spinal cord, we still couldn't get the kind of locomotion that we're used to. It wouldn't be as smooth and coordinated as what we're used to. Built into our motor system are feedback, are control mechanisms beyond the cerebral cortex to refine these electrical impulses, to refine the activity of these neurons, these motor neurons, even while the activity is being performed. One of these integrative systems is based in the thalamus. Now let's come back and look at this slice. We've seen some of these parts before. So here's my cerebral cortex, again, with the gray matter on the outside, the white matter on the inside. We can follow that white matter down as our internal capsule. And you'll notice on these slices of the brain that the inside of the brain is open, it's hollow. You might remember in cross-section of the spinal cord, we have a little opening, the central canal. That central canal is continued through the ventricles, as they're called, in the brain. Out in the telencephalon, the two lateral parts where the cerebral cortex is located, we have what we call simply the lateral ventricles. They're what indicated here by the V's. And then there are two other ventricles in the brain. So here's one lateral ventricle and the other lateral ventricle. This counts as 1 and 2. So below these here at this level, we have the third ventricle. Okay. Well, here's the reference I want you to lock into your brain. Wedged between the internal capsule here, right here, and the third ventricle, this mass is the thalamus. The thalamus is wedged between the internal capsule and the third ventricle. And we can always spot the third ventricle because it's midline. It's not paired like the first and the second. If we look at it in section here, the thalamus is often described as kind of like a big medicine pill. And I just kind of like this cute diagram at the bottom. The point it's trying to make is that the thalamus connects to everything. The thalamus is the major upper relay center in our brain. So almost everything ascending and descending through the brain passes through the thalamus. If we go to the thalamus, which is what's pictured at the top of this slide, we can color code all the different parts of the thalamus. The thalamus is really just a collection of neurons, so a collection of nuclei. Each one of them is named. If we map out where these nuclei are going, you can see that map color-coded on the sections of the brain shown below. Here we're really looking at projections onto the cerebral cortex to the thalamus. What I want to pay attention to here is this portion of the thalamus indicated by VL, and VL stands for the ventrolateral nucleus. And the reason I want to pay attention to it, look at the color coding, VL projects right here to our primary motor cortex. So we have this relationship where the nuclei in the thalamus project up to the primary motor cortex. Well, the interesting thing is the motor cortex projects down to the thalamus as well. So this is just kind of a funky, I didn't make these colors, I just kind of found this, but I liked it. So we have on the left of your image 3 different nuclei in the thalamus. We have on the right different parts of the frontal cortex. But here's the key I want you to see, if you can brace yourself and actually look at these colors. In each one of these systems, the cortex is projecting down to the thalamus, and the thalamus is projecting right back up. Here's the rostral motor. This is the premotor we were looking at. It projects down to a nuclei in the thalamus, projects right back up, projects down to VL that we were just looking at. So here, the primary motor is projecting to VL. VL projects right back. So we get essentially this little loop. Those primary motor neurons we were looking at will send a little branch down to the thalamus, to VL, activate VL. VL in turn fires right back to the cortex. And the interesting thing about this, and this little loop as we call it, so there's a corticothalamic. Our track names are really easy because we just tell you what it is. It's coming from the cortex going to the thalamus. And the thalamocortical, it's just the reverse. These are both excitatory. So the axons running from the cortex to the thalamus excite it, stimulate the parakaryones in the thalamus. They, in turn, fire back stimulating messages to the cortex. Let's come back. Here's my cerebral cortex. Gray matter, white matter. My white matter, again, collects up to form the internal capsule. Here's my third ventricle. So the mass between the internal capsule and the third ventricle that pushes into it is the thalamus. On the outside, the lateral side of my internal capsule, I have a group of parakaryones here that we call the basal ganglia. There's another part of the basal ganglia that occurs on the inside, the medial side of the internal capsule, but it pushes into the lateral ventricle, not the third ventricle. So the basal ganglia is effectively split by the internal capsule. There's one other part of what we'll call the basal ganglia that we need to come back and reference, and it sits down in the midbrain. Remember, our main landmark for the midbrain has been the red nucleus. We've got our cerebral peduncle here with that corticospinal tract making up about the middle third of it. Between the corticospinal tract and the red nucleus, this big mass is our substantia nigra, the black body of the brain. The substantia nigra works as part of the basal ganglia complex. All right, now there's a whole lot of little names and terms here. We don't need to worry about those right now. Over here is a schematic of what I just showed you. So here's my internal capsule coming down. Here's my thalamus. Here's my basal ganglia. You can see all the individual parts of the basal ganglia are labeled here. All the names are down here. I don't care about that. Here in the midbrain's my substantia nigra. We have to plug it in right down here. Here's what I care about. Here's my primary motor cortex again. It projects down into this basal ganglia complex. It's excitatory. It feeds into this network of interactions within the basal ganglia. And if you follow this, the main output from all of this interaction is right here. The basal ganglia goes to the thalamus. We already know this black line, we just talked about it. The thalamus is excitatory here to the cortex, right back to motor. But the basal ganglia, in red, is inhibitory. So follow along with me for a moment. What's not shown here in this diagram is that the primary motor cortex is projecting to the thalamus, it's projecting back, but in addition to projecting to the thalamus, it's projecting to the basal ganglia. The difference is the output of the basal ganglia is always inhibitory. So now what's happening is we have fibers going from the cortex. The basal ganglia is also called the striatum. So we have a corticostriate fibers. They excite the basal ganglia from the cortex. But the basal ganglia doesn't play that game. It sends inhibitory signals to the thalamus. Thalamus is normally all excitatory. What the basal ganglia can do is dial that down. So the basal ganglia can modulate the interaction between the cortex and the thalamus. We're back here at the cerebral cortex, giving rise to our tracts of white matter that come together to form the internal capsule. Here's our nice map of the internal capsule, which didn't make perhaps much sense before, but now look at it. Here's the internal capsule coming down. On one side of it is the thalamus. On the other side are these parts of the basal ganglia. My third ventricle would be right here, and up superior is the other part of the basal ganglia that would be projecting into the lateral ventricle. Most of the internal capsule, if we map out the internal capsule, most of it would be made up by a group of fibers that are called the corticopontine fibers. The majority of the output of the cerebral cortex is what is called the corticopontine tract. I'm going to come back to the corticopontine tract for just a moment, but I have to prime my story. Here we're looking at the sagittal brain. Right? My thalamus is sitting right in here, my midbrain right in here. Here's my big swollen pons. Here's my medulla. We can kind of almost make up where the ventricles are. Superior to the pons, I have the fourth ventricle of the brain. We've been talking about the third ventricle. Here's the fourth. And forming the roof of the fourth ventricle is the cerebellum. If we look at a little section through the cerebellum, we see that it has a cortex that in some ways is very similar to the cerebral cortex. It's folded in. We don't call them gyri and sulci, but it's the same arrangement. The parakaryones are on the outside, the gray matter's on the outside, and then we have this mass of axons, the white matter, radiating down into primarily the pons, but also the midbrain and the medulla. All right, what about our corticopontine? Let's come back. Here's our pons in section, and again, the dominant part of the pons is this big, swollen, think of it as the beer belly of the pons. That big swollen mass is called the basis pontis. And the reason it's swollen is because it is full of what are called pontine nuclei. This is where most of the fibers of the cerebral cortex are running. So the corticopontine fibers are coming down that peduncle and they're synapsing on these pontine nuclei. What do the pontine nuclei do? Their axons are running across. You can see them right here. Their axons are running across and they form this big mass that is called the cerebellar peduncle. We saw a cerebral peduncle earlier with the corticospinal tract, its middle third. That cerebral peduncle, again, was an expression of the internal capsule and the axons running, connecting the brain stem to the cerebral cortex. The cerebellar peduncle is an expression of all of the axons connecting the pons, the medulla, and the midbrain, but especially the pons up to the cerebellum. That peduncle is going to lead to the white matter on the inside of the cerebellum we were just looking at. So that mass of fibers we refer to as the pontocerebellar tract. You'll notice it's sitting up above, just superior to our corticospinal and rubrospinal tracts here. All right, so we have a big mass of fibers coming down from the cerebral cortex, synapsing in the basis pontis on the pontine nuclei, and then going from there as the pontocerebellar tract into the white matter and ultimately into the cerebellar cortex. I need one more player here in my story. I am back in the medulla, and I can tell I'm in the medulla because I have this big swollen ventral surface, the pyramid, but just superior and lateral to it, I have this big body here that is called the inferior olivary nucleus, or more commonly, the inferior olivary body. The inferior olivary body is the primary relay center for the lower part of the brain. It's the inferior counterpart to the thalamus. It's not as big a relay center, but it is a dominant relay center. It's getting input from the spinal cord as well as from our cerebral cortex. And what does it do to it? It sends it predominantly up to the cerebellum. So if we just look at the cerebellum in a simple form for a moment, it gets really four forms of input. It gets input directly from the spine. These are mainly from sense organs in the body, especially the muscle coming into the cerebellum. It gets input from our vestibular system in our ear, telling us where our body is in space. But then its biggest input is the pontocerebellar tract and the olivocerebellar tract. So we have these four tracts running in these cerebellar peduncles going into the white matter of the cerebellum. How does the cerebellum work? Well, it's a two-level balancing act. And here's what I mean by that. All of those inputs I just mentioned to you, all of those inputs come into the white matter, and at the bottom of the white matter, there's four nuclei, four clusters of parakaryones that are called the deep cerebellar nuclei. All of these inputs send excitatory signals into at least one of those deep cerebellar nuclei. So the inputs are all excitatory. Then they go up the white matter of the cerebellum. They go out into the gray matter of the cerebellar cortex. And here they do all sorts of wild synaptic circuitry. We don't need to worry about that. But ultimately, the signal ends up in a special neuron that's only found in the cerebellar cortex called a Purkinje neuron. And a Purkinje neuron does just one thing when it's activated. It sends an inhibitory axon down onto the deep cerebellar nuclei. So the deep cerebellar nuclei always get excited by the input fibers and inhibited by the Purkinje neurons. What do the deep cerebellar nuclei do with the balance? Whether it's inhibitory or excitatory? Well, they give us our cerebellar output. And that cerebellar output, there's three that we really care about. One, it runs right back to the thalamus. And of course, from the thalamus, it goes right up to the cerebral cortex. So ultimately, we could really think of this as a cerebellar cortical tract. It also goes from the cerebellum back to the red nucleus, and it goes from the cerebellum to the reticular. In this way, what the cerebellum is doing is it refines ongoing muscle activity, so it smooths out the muscle interactions. It also functions in planning. This circuit will start before the muscles are actually firing. All right, so what do we have when we put this together? Well, most importantly, we have to get past this. We tend to have a very simple view of the nervous system, and even when we talk about it, even when we teach it, we tend to pretend that it's like one cell in the cerebellar cortex, one little neuron that runs down to a muscle. It's not that. If we want to have a more realistic view of it, we have to look at it like this. The cerebral cortex projecting not only to the spinal cord, but to the red nucleus, the superior olive, the reticular formation, the thalamus, the basal ganglia, and to the cerebellum. Thalamus in turn feeding back, the basal ganglia influencing that, the red nucleus going to the spinal cord, as does the olive, as does the reticular formation. The olive going to the cerebellum as well. Then the cerebellar output going back to the thalamus, the red nucleus, and the reticular formation. This is a much more realistic level, especially when we add back in the little circuits by the interneurons of the spinal cord. So if we really want to understand how we walk down a sidewalk, this is kind of the level we need to look at. And you may think that this is just kind of overdoing it, and we're just having fun with the anatomy, but you know, I set this figure up on purpose. So we have here the red nucleus, the basal ganglia, and the cerebellum. These three have an important thing in common. They are so important in integrating motor signals that a pathology in any one of these three is associated with abnormal motor movements and especially tremor. Red nucleus pathology produces a very characteristic tremor that's called rubral tremor, the red tremor. Cerebellar tremors are especially interesting. We get what are called intention tremors. The tremor starts before the movement. Again, think of the cerebellar role in planning. And then, of course, we have the basal ganglia. The basal ganglia includes that substantia nigra, which is the main center of dopamine in our brain. Abnormal levels of dopamine change the function of the basal ganglia. They can make it too inhibitory or not inhibitory enough. We end up then on a spectrum of motor disorders ranging from Parkinson's disease to things like Huntington's chorea. So the action of these three, and we'll do this in much more detail, but the action of these three show us quite clearly how important integration of this system really is to normal function. This is where I want you, at least initially, to be thinking of the motor system, but please bear in mind, this is woefully still simplistic. I haven't added in any sensory component. We have coming up from our spinal cord all sorts of sensory information about the position of our joints, the contractile state of our muscles. That sensory information is going to project up through the olive, the red nucleus, the thalamus, the reticular, and especially the cerebellum. And that sensory input is going to alter the output and the relays back to the motor system. We really can't understand one without the other, but that's getting more detail than I wanted to give you in this introductory lecture. I hope this helps you. Thank you.